Today's talk is based on work that has been done in conjunction with my working group. This project involved about half the people in the working group, maybe a little bit more. In particular, you'll notice a familiar name there. Carolyn joined our working group. Were you at our first meeting? I think so. Yeah, between when we got approved for a working group and we actually had our first meeting, we brought Carolyn aboard. And also Angie Peace, who was a postdoc here last year, is part of our working group. And both of them, along with my co-organizers, Roger and Sabrina, and Roger's colleagues, Eric and Elka, contributed to the material that I'm presenting here. I always start every talk that I give on plants with the thing that got me interested in plants in the first place. Sabrina Russo and I were co-mentoring a team of undergraduate math and biology students, and at our first organizational meeting, Sabrina showed this picture. So this is a combination of a soil map and a tree census. So the background colors are the soil map. White is sandy soil, dark green is clay soil. The other two colors are something in between. The circles and dots are individual trees. This is at a national park in Borneo. Every single tree has been censused. This area is very thoroughly studied. And so you can see that the Aromatica tree lives in the sandy soil, but you don't see any of them on the clay soil and hardly any on the two intermediate types of soil. And Lanceolata lives almost entirely on the clay soil and hardly anywhere else. They're in the same genus, so these species are not that much different. But small variations in soil type give local advantages to different species. And what's particularly interesting is that the life history strategies are very different for these different soil types. Lanceolata grows very quickly. It builds flimsy leaves that don't last very long by tropical forest standards. Aromatica, as its name suggests, invests some of its resources in chemical defenses for its leaves. It grows very slowly. It builds sturdy leaves that are suffused with camphor. Dryabalonops aromatica is the camphor tree. And so this is what originally gave rise to the project that Sabrina and I developed into the NIMBioS working group. Our main long-term goal is to understand complex trade-offs in function, functional integration, and adaptive responses to environmental heterogeneity. What I particularly like about her tropical forest study is that there's only one dimension to the environmental heterogeneity. The rainfall is the same, the temperature is the same, the humidity in the air is the same. It's only a 1 square mile or so area. The only thing that's different is the soil type. We want to look at connecting tree life history strategies to these different environmental conditions. Can we build a model that will predict that the sandy soil will support a tree like Lanceolata and the clay soil will support a tree like Aromatica? And we want to explore species diversity in response to climate change. Our medium-term goal is what we're pursuing in the NIMBioS working group, and that is to develop a dynamic energy budget model for growth of a single tree. So here's a schematic of what a dynamic energy budget model looks like. This is taken from a survey review, not sure what to call it, paper that I published about a year ago. So in a general animal version of dynamic energy budget model, we're tracking the resources. Resources are collected as food. They get absorbed and there's some loss in feces. What gets absorbed goes into a reserve. From the reserve, it gets mobilized and then allocated in one of two streams. The left side is the somatic stream with growth-related tissues. There's some loss for maintenance, and then the resources get used to build more structure. Structure being a euphemism for anything in an animal that contributes to getting food. So that includes your muscles, your bones, your digestive organs, just about everything. The other stream is the reproductive stream, and there's this abstract box for maturity, which isn't really a physical entity, but dynamic energy modeling is kind of abstract. And when the maturity box gets full, then whatever was going into the maturity stream gets sent to reproduction instead. And so that's what the basic dynamic energy budget model looks like. It's simple for animals because they collect all their resources in the same way. So you only need one structure and one reserve. But that's not the case for plants. Plants need at least two structures and two reserves for carbon and nitrogen. And so that introduces a whole lot of complications, like, for example, there's an additional allocation needed. So in this area, we're now going to have to allocate not only towards reproduction and somatic growth, but now the part that goes to somatic growth is going to have to be allocated between growth structures. And that's going to have to be, you're going to have to be able to make dynamic changes to that. So if you're a temperate tree, and the winter comes along and you drop your leaves, then at the beginning of the spring, you're in a very different situation than you are in July. And so the strategy that you follow at that point has to be based on what your current status is. So what model elements would you need for a plant? Well, you need state variables that track the sizes of various organs over time. So mass of leaves, mass of assimilative roots at minimum. And we also include transport structures as a separate state variable. You need layout or architecture variables that track the geometry of the tree. For example, the crown radius, the total leaf area as opposed to the total leaf mass, the leaf area to sapwood area ratio, things like that. This is part of Carolyn's expertise, and that's why we wanted her to join our group. You need resource variables that measure the resource availability. So how much nitrogen is there in the soil? That's a lot more complicated than it is for animals or for light. So for animals, for a simple dynamic energy budget model, you can just assume the food supply is constant, that the animals eat something that whose population is not depleted by the animal doing the eating. But you certainly can't do that with plants in soil. However much nitrogen plants remove from the soil, that nitrogen is no longer available. So there's a feedback that you have to keep track of. And then we need uptake rates, which are variables that measure the current rates of resource acquisition. So for example, what's the current nitrogen flux from soil to root? Here's a schematic of what the structure of the model will look like. And in the dynamic energy budget model for animals, it's really all here. What corresponds to our plant physiology model is usually very simple in an animal model. You can just assume that the rate of uptake is proportional to the size of the structure. You can't do that with plants. The amount of nitrogen uptake is not just proportional to the amount of roots that you have. It also depends on how much nitrogen there is in the soil and other things. So we need a separate physiology model, which is where I've been putting my work in during my sabbatical fellowship here. And that interacts with the resources in the soil. And then we're a bit behind on doing the architecture work because you can leave that out and make some simple assumptions and still get a model that has some limited value. So now let's just look at the growth submodel. So in today's talk, I'm going to talk about the dynamic energy budget part. So this is within the growth box. We have carbon coming in and nitrogen coming in from different directions, and those resources are being distributed, allocated among leaves, stems, and roots in some manner. And so the model needs rules for deciding how to direct these input streams. In the standard DEB dogma for animal models, it is always assumed that there is a fixed fraction of allocation that goes to maturity/reproduction. I'm very skeptical of that in the animal model, and I'm even more skeptical of that in the tree model. So we're assuming that the maturity reproduction stream only gets turned on after the plant reaches a certain development level, and so we're not sending any of the resources in that direction for younger plants, which is all that we're working on right now. So our allocation is simply between structures that go to collect carbon and structures that go to collect nitrogen. And there's some considerations. Each organ has a particular stoichiometry. So even if you think of leaves as a single organ, when in fact you could divide it up into several smaller organs, there's a nitrogen-carbon ratio used to build leaves. That might be different from one plant to another. It might be different from one leaf to another, but we're going to ignore that for now. But it's certainly different between leaves and roots. One resource may be more limiting than the other. If things are in stasis, if the environment hasn't changed a lot recently, you can expect that plants will operate so that the two resources are equally limiting. If you're living in a low nitrogen environment, then you build more roots so that the nitrogen is not more limiting than carbon. But things can happen that can make one resource more limiting, like the leaves falling in the autumn in the temperate zone, or other sorts of things can happen. So at times, one resource may be more limiting than the other. Transport capacity needs to be adequate. We're not going to deal with that in the current model that I'm talking about today, but that is a significant issue for determining allocation rules that we're still working on. And allocation must be able to respond to current needs. So if you lose your leaves, the allocation model has to be capable of directing the tree to work on building leaves and not building roots. Okay, so back to the project goals. That's an overview of our medium-term goal, the goal of the NIMBioS working group. The short-term goal that I'm talking about today is developing a dynamic energy budget model that merely predicts how resources are allocated between roots and shoots. We're going to ignore transport capacity. We're just going to assume that some fixed proportion of roots and some fixed proportion of shoots are transport, and we'll just not worry about it. But we want to find a simple way of deciding how resources are allocated between roots and shoots. So the key theoretical question is, how do plants allocate resources between roots and shoots? There's a standard mathematical answer that comes from a seminal paper by Iwasa and Roughgarden, which I think was Iwasa's PhD thesis. And that is, you grow whichever organ is deficient until optimal proportions are achieved, and then grow both to maintain the balance. So optimal proportions could be something that varies over time. It could be that for a small plant you want one root-shoot ratio, and as the plant grows, maybe that root-shoot ratio that's optimal should change. But at any given size, there would be theoretically an optimal ratio, and the plant should try to maintain that. Okay, that's a mathematical answer. The theoretical problem is that optimality arguments assume some capability for central planning. So it could be plants have some capability for central planning. They certainly don't have any sort of central nervous system that can do it. But it could be that they have some kind of hormonal feedback that makes the roots grow at the right time and makes the shoots grow at the right time. In any case, it's not clear that this is biologically realistic, and it's also not clear that it's theoretically necessary to have any sort of central planning. And that's the issue that we're dealing with in this model. So we are examining what we call the local control hypothesis, that allocation according to local rules operating independently in each organ achieves near optimal results. So we're not going to assume that there are any hormones that determine whether we grow roots or shoots at the moment. We're going to assume that the roots don't know what the shoots are doing and the shoots don't know what the roots are doing, and it all somehow comes out right. So in order to get a sense of how that might work, let's first talk about obligate sentraphy in microbes. So some microbes are unable to produce essential metabolites, but they overproduce others. The species that do this occur in pairs that supply each other's missing metabolites. This is actually common in nature. And experiments have shown that there are some cases where the fitness cost of overproducing compound A might be less than the fitness gain for not having to produce compound B. So if you could just find a partner, it might be in your self-interest to produce more of one essential resource and not have to build any machinery to produce another one, but count on being able to trade your excess resource with your partner. So how do you share resources without central planning? So in the case of pure obligate sentraphy in microbes, there's obviously no central planning. How do you do it? Well, each symbiont uses as much of the resources it makes as it can possibly use, and it excretes the excess. So if I'm making a huge amount of resource A and I'm hardly getting any resource B from my partner, I can't use much of the resource A that I produce, so I excrete the excess, which is then available to my partner. Each symbiont relies on its partner's excreted excess for the resource it can't produce for itself. And selfish independent allocation allows each symbiont to prosper. So one of the one of the things that led to this idea was that one of our team members, Eric Muller, had done a paper on dynamic energy budget modeling in corals, which is one example of a syntrophic relationship. And so the question is, can we model trees as obligate syntrophs? Well, let's talk about an idealized pre-reproductive plant that consists of two potentially obligate syntrophs. So we're going to have two organs. We'll euphemistically call them roots and shoots. Roots collect nitrogen and shoots collect carbon. I've got all these things in quotation marks because they do other things. So shoots also collect light, roots collect phosphorus and water. Roots and shoots are really differentiated into sub-organs, but we're going to ignore all that. Our idealized plant is simply going to have roots and shoots, each of which has only one function. We'll have variables for the root biomass and the shoot biomass. For convenience, we'll measure them in C moles, moles of carbon. We need two synthesizing units that create roots and shoots from carbon and nitrogen. Synthesizing unit is a term that we've lifted from the dynamic energy budget literature. It essentially, think of it as a factory that takes resources and produces tissues. So it's an abstract way of thinking about how resources combine together to make new tissues. So 1 C-mole of organ J requires 1 mole of carbon to produce, plus whatever is lost in the reaction. It also requires some amount of nitrogen, and this has to be organ-specific. So the roots have a certain value of beta sub r, for how much nitrogen they need to produce 1 unit of roots. And then there's a beta sub s for how much nitrogen is needed to produce 1 C-mole of shoots. And so these are two different values. And what's critical is the ratio between the two. So if you divide the nitrogen ratio of shoots by the nitrogen ratio of roots, you get a measure of the relative importance of nitrogen to shoots versus roots. That's also a measure of the relative importance of carbon in roots versus shoots. So I like to think of beta as the relative importance of the imported resource to the locally produced resource. If beta is bigger than 1, it means you need your partner's resource more than you need your own, regardless of which of the two partners you are. And if beta is less than 1, it means you need your own resource more than you need your partner's. Okay, we all know that leaves have a lot of nitrogen in them. And so there's some reason for believing that beta is greater than 1, even though shoots is more than just leaves. It also includes the stems, which don't have much nitrogen. But then the stems in the roots don't have much nitrogen either. So it's certainly reasonable to think that beta is bigger than 1 for plants, that the leaves need more nitrogen than the roots do. We need processes of growth and tissue loss, different types of tissue loss. And to make this more sophisticated, this isn't really essential, but we're going to allow some reabsorption of lost nitrogen. Plants are really amazing in the way they function. And one of the things that they do is they reuse nitrogen. They're very protective of their nitrogen. When you see leaves turn color in the fall, it's not because the leaf is dying. The leaf dies later, after it turns color. What you're actually seeing is the tree pulling resources out of the leaves. And the resources that make the leaves green are being removed from the leaf. And similarly, other various nitrogen-containing compounds are being removed from the leaves and stored as much as possible. And we need uptake processes, which will include photosynthesis as well as nitrogen uptake. But we're going to abstract the uptake processes in order to focus on the allocation strategies. There's no further differentiation of roots and shoots, and we're not going to include water or other nutrients. So here's a schematic that shows the variables and resource flows that need to be built into the model. So R is the root biomass in C moles, and S is the shoot biomass in C moles. Each one has an arrow coming in. I've never had the luxury of a pointer before, so I tend to just want to walk over there and point with my finger. So we've got these resources coming in. QR and QS are the biomass production rates. The synthesizing unit is this dot here. So this dot is a synthesizing unit that takes carbon and nitrogen and produces root tissue. And Q sub R is the rate at which root tissue is produced. Q sub S is the rate at which shoot tissue is produced. Those rates are increasing, S and R, and then the T's are the rates of losses that are decreasing, S and R. So we'll build a differential equation for S, for example, that has Q as an input and T as an output. The U's are resource production rates. They go into the synthesizing units. Oops. So they go into the synthesizing unit. And because we're assuming local allocation, all of the carbon that's produced by the leaves goes into the shoot synthesizing unit. None of it goes into the root synthesizing unit. Whatever carbon the roots are going to get is going to have to be rejected by the shoots. It's not going to come directly from the leaves. There is reabsorbed nitrogen from old roots and shoots, and so in the case of roots, that just gets recycled back to the root synthesizing unit. In the case of shoots, there is a natural supply of nitrogen that doesn't have to come from the roots, but it's a small supply. So if you're not getting any nitrogen from the roots, you still have a little bit of nitrogen coming from the shoots, but it's only coming from reabsorption from old leaves that you're about to drop. Each synthesizing unit potentially has an excess of its locally produced resource. So here the root synthesizing unit could possibly have an excess of nitrogen, and that excess gets rejected and goes to the shoot synthesizing unit. Similarly, the carbon stream coming into the root is the same as the rejection flux coming out of the shoots. It's possible that the shoot synthesizing unit could have more nitrogen than it needs. I mean, this isn't likely to happen often, but it could happen, like if all the leaves have been stripped off. And so it's possible that there is an excess of nitrogen in the shoot SU. In our simple model, we're assuming that that's lost. You could assume that it goes into storage or something else, but we're assuming that it's just lost, and it isn't included in this diagram, just to keep the diagram relatively simple. All right, we need some physiological assumptions. These are as simple as possible. Some of them are realistic, some of them are overly simplified. So tissue loss rates are proportional to organ total amounts. That seems reasonable. Reabsorption rates are proportional to tissue loss rates. That seems reasonable. Uptake of external resources is proportional to organ total amounts. This is not a very good assumption, but it will suffice for the model that we're doing. So the problem with this is that it doesn't show any effect, any density dependence. What actually happens as a tree gets bigger is that the leaves start to the new leaves start to shade the old leaves. And so if you double the size of a tree, you don't double the amount of carbon that you can collect from the atmosphere. You don't do quite that well. But we're ignoring that for this simple model, and I don't think it makes a very important difference. One technical note is that there's no reason to consider U sub n and R sub r separately. They're both sources of nitrogen available to the root SU, so why not just add them together? And so wherever they appear as the sum, and I'm just going to replace them with a convenient algebraic form. And so that introduces this new parameter alpha n that replaces alpha n prime. And so then we can eliminate the, so essentially what we're doing is we're making this 0, and we're increasing alpha n to become some alpha n prime to become some alpha n. Next is the resource economy in the root SU. The rejection fluxes are calculated by mass balance. The flux out is the flux in minus the consumption. So there's a carbon flux into the roots, which is strictly coming from the shoot SU. There's a carbon consumption rate, which is what is produced by the synthesizing unit. So in this model, we're ignoring the fact that there is some waste in chemical reactions. There will be some excess carbon dioxide perhaps that gets lost. We're going to ignore that. And then there's a carbon flux out that's lost to the system because this is the root synthesizing unit. Nitrogen, there's a flux in from root uptake and reabsorption, a consumption for new roots, and a flux out to the shoot SU. And so using this simple balance that flux out is flux in minus consumption, you can write down formulas for the two fluxes out in terms of the flux in and consumption. The W flux doesn't matter. I'm just writing that down for completeness, but we're not going to use it. But the formula for rho sub n is important because that will tell you how much nitrogen is available to the shoots. And so similarly, there's a resource economy in the shoot SU. I don't think we need to spend any more time talking about that. All right, so from the diagram, the dynamic variables have growth and tissue loss, so you get a differential equation. The overall rate of change of roots is the rate they're produced minus the rate at which they're lost, and same for shoots. Growth rates are defined by some synthesizing unit model to be specified later. So in order to figure out what the growth rate is for roots, you need some function in which you input the amount of roots that you can produce using the local resource, and then the amount of roots that you can produce using the imported resource. For the shoots, the inputs are the amount of shoots you can produce using the local resource and the amount of shoots you can produce using the imported resource. So in both cases, the local resource is listed first. That way we can use the same formula for the synthesizing unit. And so these equations, together with the equations that determine the rejection fluxes, are a complete model. So 1, 2, and 3 are a set of 6 equations for R and S, QR and QS, rho N, and rho C. And so we just have to specify an F and solve the set of equations. So I'm not going to go into a lot of details about the mathematics. I want to focus on what the results say and what they mean. But we do need to talk about some assumptions about synthesizing units. We're going to look at 2 different synthesizing units. First, we're going to look at the minimum rule synthesizing unit model. This occurs in the literature a lot. We'll assume that synthesizing units are perfectly efficient so that whichever resource is limiting is completely used up and there's no excess. This is the Liebig minimum rule. So this says you take whichever resources will produce a smaller amount of that tissue, and that's how much of that tissue you get. And if you substitute the equations together as much as possible, you get a system of functional equations for the 2 growth rates. So there's an equation that expresses Q sub R in terms of Q sub S, and Q sub S in terms of Q sub R. So in order to calculate Q sub R as the minimum of what you can make from roots, from nitrogen and what you can make from carbon, you have to know how much carbon you have available. And in order to know how much carbon you have available, you have to know how much shoots you're producing. So these 2 equations are coupled. And we have to solve this system by considering cases because we have the minimum function. And what you get depends on whether beta is greater than 1 or less than 1. So first we're going to look at the case that we think is more biologically realistic nearly all the time, if not all the time. The case where shoots need more nitrogen than roots do. And so in that case, you get 3 different cases that you have to solve, depending on which of the 2 choices is the minimum in each of the 2 equations. So there should be 4 cases, because you've got an A or B for the first equation and an A or B for the second. But it turns out that one of those 4 combinations is impossible, so there's really only 3. And it's convenient to plot these in a graph like this. So you need to know what is the shoot-root ratio, what is S divided by R, and you need to know what is the R growth rate, Q sub R, and this is the growth rate relative to what it could be if you put all of your resources into roots. So it's scaled from 0 to 1. It's not the actual growth rate, it's the fraction of the growth rate that is actually achieved. So it'll be 1 if you're putting all of your resources into growing roots, and 0 if you're putting all of your resources into growing shoots. And which of those you have, or something in between, depends on the current shoot-root ratio. So the growth history is a composite of these three cases, always leading to the optimal steady state, which is marked in the graph. So let's look at the three cases. Case number one, the carbon is deficient. If carbon is deficient, the shoot-root ratio is low. What makes the carbon deficient is the fact that the shoot-root ratio is low. So you have a low ratio of shoots to roots. You have inadequate carbon. And so there's no root growth. All of the carbon is being used by the shoots. There is no rejection flux down to the roots. So the roots don't grow at all. And so you're at 0 on this graph. In case 2, it's the opposite. Now you have a really large shoot-root ratio. So you have lots of carbon compared to the amount of nitrogen you have. The amount of nitrogen you have is deficient. And so there will be no shoot growth in this model because all of the nitrogen that's available is being used by the roots. The shoots have a small amount of nitrogen that's available from recycling, but it's not enough to maintain the amount of shoots that you currently have. So you're losing shoots at a current rate because they're getting old, and you're only harvesting the nitrogen, a portion of the nitrogen that you lost. So you can replace some of that, but not all of it. So the net growth rate of shoots is going to be negative. We'll just call it 0 for the purpose of this graph. In the intermediate case, where you have a medium shoot-root ratio, then both structures grow. So because beta is greater than 1, it means that the shoots are deficient in nitrogen, since they need more nitrogen, and the roots are deficient in carbon, since they need more carbon. So both structures will grow. Each one has more of its locally produced resource than it needs. So each one sends its excess of its locally produced resource to its partner, and so that partner can grow. And so you get both growth of roots and shoots. Which one grows more depends on where you are compared to the steady state. If you have a pretty high shoot-root ratio, but not quite over the critical value, then that means that you have a lot of excess carbon going down to the roots, only a tiny amount of excess nitrogen going up to the shoots, so you're not going to produce much in the way of shoots, and you're going to produce almost as much roots as you could. So here's an example over time of what would happen in this case. Let's say you start with a low shoot-root ratio. So it's the springtime now in the temperate zone, and so at time 0 there's not much in the way of shoots, meaning leaves. And so the whole tree is C-limited for a while. That means only shoots will be built. In the shoot synthesizing unit, there's more excess nitrogen coming up from the roots than is necessary to build shoot tissue along with the small amount of carbon that you have available. So the whole tree is C-limited, only shoots are built, and the roots slowly atrophy. And this will hold for a while until the optimal ratio is achieved. And when you get past that, then the roots are C-limited and the shoots are N-limited. And so you get this nice balance in which each one is producing more of its local resource than it can consume and sending excess local resource to its partner so that both partners will be growing. The solution approaches steady state, and you can show that this growth history is optimal. So it's not only approximately optimal, it is optimal. So what that means is that if you have two plants, one of which follows this local allocation rule, and another one that has an arbitrary allocation rule in which the fraction of nitrogen that gets rejected is not rho sub n, but it's whatever you want it to be. It turns out you can't do any better than to make the same thing that the local allocation rule gives. So at least in this version of the model, in this scenario, you don't need any kind of communication between roots and shoots in order to get optimal behavior. All you need is for the shoots to be more N-limited and the roots to be more C-limited. The case is quite different. The situation is quite different if beta is less than 1. Now each of the components needs more of its local resource than it needs of the imported resource. So that means that we still have the same three cases as before, but the middle case, the line now has the opposite slope. And so there's still an optimal strategy, but you can't get there. So there's no logical progression towards the optimal steady state. If you start out down here, you can't get to there. You can't walk up this graph. So what actually happens? Well, my colleagues and I are still debating what actually happens, and so I'm giving you my version of what actually happens. So what I think actually happens is, let's say the initial shoot-root ratio is 0.2. So we're down here somewhere, just like we were before. The system starts in case 1. All the resources are used for shoots. So there is no carbon going downstairs. It's all being used at the shoot SU. And once you get to this point where there are multiple solutions, there's no reason why the system should switch to a different solution. The shoot SU is using all the carbon it can use. Why should it stop? According to the local allocation system, if it can use all the carbon, it will. And so we'll continue along this horizontal line until we can no longer use all of the carbon that we're producing, and that will happen at a much higher shoot-root ratio. So we'll go along here, and once we get there, then there's a problem. Now the whole tree is N-limited, so shoots cannot grow. And so since the shoots can't grow, that means suddenly all of the carbon production gets exported down to the roots. And so control of the tree is now changed. In an instant, control of the tree is passed from the shoots to the roots. Now the roots are controlling the tree, and so we'll suddenly jump from here to here. And now we have the reverse situation. The roots will be using all the nitrogen that they can, and they will do so for as long as they can. They will hoard nitrogen for as long as it's possible to do that, which will be to this point, and then the cycle will start anew. So if you look at how this would work over time, you have these alternating periods in which one part of the tree is dominating and the other part is losing out. And this time you're getting suboptimal results. So the optimal result would be you start out with all shoot growth because we were short of shoots, but at some point it would be better to split your production between shoots and roots, but you're not actually doing that. So you'll actually do better for a very brief moment if you continue to put everything into shoots, but not for long, because eventually the control will transfer over to the roots, and now the shoot amount stays the same for a while the roots grow. Meanwhile, if you were following an optimal strategy, both would continue to grow. And so you would clearly do better if you chose a balanced strategy rather than this back and forth between, or this bang-bang strategy between one dominating and the other dominating. So the shoot-root ratio never reaches a steady state value. The cyan curve shows the optimal behavior in which you would reach a steady state value and then maintain it. This doesn't seem to happen in nature, at least not in any trees that my biologist colleagues have seen. And it could be that it doesn't happen because this is a flaw in the model, which I think is part of the reason. It could also be that it doesn't happen because beta is really greater than 1, so this scenario never arises, which I think is also true. All right, so I think that one of the things that this shows is a flaw in the minimum model. The minimum model is really unrealistic, and so we could choose a more realistic synthesizing unit model based on chemical kinetics. So if we use a derivation based on chemical kinetics, we get a more complicated equation. It's no longer the minimum of A and B, it's now some function of the two, which we can rewrite in terms of the shoot-root ratio. Again, let's not worry about the mathematics, but let's think about how this synthesizing unit will function. So for the minimum rule, if B is less than A, the minimum rule says you choose the smaller one, which is B. So as long as B over A is less than 1, then Q over A is equal to B over A, so the minimum rule gives that line. If B is bigger than A, then the minimum rule gives Q equals A, so Q over A equals 1. So that's why the minimum rule graph looks like this. That's the graph that we've been using for how Q over A compares to B over A, whereas the chemical kinetic synthesizing unit, or co-limitation synthesizing unit, gives this. So it approaches the minimum rule for ratios greater than 4:1, but it's noticeably different for near equal resource levels, and I was surprised by how much different it is. So if you take equal resource levels, this value is only 2/3 of that value. It's quite a lot less. So if you have a good balance between the reactants in a chemical kinetics-based synthesizing unit model, you actually have a fair amount of rejection flux of both of the reactants. And that's because the reactions are not instantaneous. It takes some time. You can't connect the carbon and the nitrogen together as fast as you can receive them. All right, so here are some results from the chemical kinetics synthesizing unit. So these graphs correspond to the previous graphs, but now with a comparison between the minimum rule and the chemical kinetics rule. And I'm only looking at the beta greater than 1 case. The beta less than 1 case doesn't show the same difference. It's similar. In the chemical kinetics, it doesn't make so much difference whether the beta is less than or greater than 1, because there's always a rejection flux regardless of which you have more of. So this shows that if you could choose between the minimum rule and the chemical kinetics rule, so if a plant could evolve both kinds of synthesizing units, and whichever was better was the one chosen by natural selection, then you would choose the minimum rule, obviously. I would argue that there's no reason to think that you could do that chemical reactions don't occur according to the minimum rule. They occur according to chemical kinetics. And so in my view, this dashed curve is an ideal that cannot be achieved in practice. So in practice, though, you get a very similar result for how the tree grows. The dashed is for the minimum rule, and the solid is for chemical kinetics. And so if you're looking at this graph, you don't really see a significant difference between the two. So the predictions are qualitatively the same, except you don't get this bizarre hysteresis when beta is less than 1. The growth is slower and the transitions are smoother. All right, now let's talk a bit about the effect of nutrient availability. This is a bit of work that Carolyn and Angie did while the rest of us were doing some other stuff. So the first graph shows what is the optimal shoot-root ratio if you change the rate at which nitrogen can be taken up. So we're guessing that that's the main reason why sand and clay are different. That the trees growing in sand have better capability of absorbing nitrogen. And so alpha sub n is bigger if you're living in a sandy soil than it is if you're living in a clay soil. So the question is, how will that change your optimal shoot-root ratio? And the model predicts what we would expect to see. That if you have better access to nitrogen, you're going to have a larger shoot-root ratio. You don't need as much roots, so you're going to put more of your resources into building shoots. Take advantage of your better nitrogen access to build more leaves and collect more carbon, and then you will do better than if you didn't do that. Also, the optimal shoot carbon content is higher. That's a fairly minor difference, but it's a noticeable difference. So if you look at how does this lambda is the growth rate. So if you ask, how does the growth rate depend on what you choose for the nitrogen content of leaves? So the horizontal axis is how much nitrogen do you put in your leaves, and the vertical axis is how fast do you grow. And the different curves are for different soils. So if you're in the sandy soil where nitrogen access is better, that's the blue curve. And so this is your optimal nitrogen content in shoots. If you're in the clay soil, your optimal nitrogen content in shoots is a little lower. Okay, so in the soil where nitrogen is more available, you put more nitrogen into the shoots. It's not a very large effect, but it's an effect that perhaps could be measurable. And it is something that we do see in the stoichiometry of trees that live in different soils. Okay, I just want to finish up with some key conclusions. Obligate sentraphy is stable when each partner has a relatively greater need for the imported resource than its partner has. That was true for both synthesizing units. And local allocation yields optimal growth in this case. It can be unstable and suboptimal when each partner's need for its own resource is relatively greater than that of the other partner, maybe. So we don't know if it actually is unstable and suboptimal, but it seems like it might be unstable and it might well be suboptimal. Modeling certainly supports the conjecture that plants do not require active coordination between roots and shoots. One thing that we've glossed over here, though, is we have not had a separate allocation rule for how much transport you need. And this same method will not work for that. If you think of stems as being intermediate between roots and shoots, and you allow them to use all of the resources they get and only excrete what is in excess, what you end up getting is that the transport structure always dominates and controls everything, and so you only build transport, you don't build any more leaves and roots. Obviously, that doesn't happen. So this local allocation mechanism does not work if you think of transport structures as living in between shoots and roots. We need some other sort of allocation rule for that. And there's probably, I'm sure we can find some allometric rules that will allow us to just decide that for every, that there's some graph of how much transport you have versus how much shoot you have, and you build transport tissue to maintain that function. And finally, obligate sentraphy would be a great name for a rock band. Okay, thank you. Any questions? Sure.
Does the diurnal nature of nitrogen-carbon cycling have any ability to allow it to switch strategies? And then also sort of like the seasonal nature of carbon as it goes through?
Seasonal certainly does. We don't think that the diurnal nature will affect this model. Plants actually do their uptake during the day and their growth largely at night, if not completely at night. So in our model, the rates of carbon and nitrogen acquisition are just proportional to the amount that you have. But in actual fact, those are both controlled to some extent by how open the stomata are. And if you're in the dark, the stomata are as closed as they possibly can, which means that there's no water transpiration through the plant. If there's no transpiration, there's no photosynthesis, because the same structure that allows the water out lets the carbon dioxide in. It's not quite so clear that cutting off flow cuts off the nitrogen intake, but it probably cuts off most of the nitrogen intake. Most of the nitrogen intake comes along with the water flow. So at night, none of this is happening, but the growth is occurring. On the other hand, the time scale of diurnal variation is pretty short, and so we don't think that will affect this. There are some things in the physiology where we cannot leave out the effects of diurnal variation. Suzanne?
So in these dynamic energy budget models, do people tend to include the effects of environmental stochasticity, or are they?
Not in models that I've seen. So you certainly could, and you should for plants. For animals, since they're getting their resources from the biosphere rather than from the abiotic environment, there isn't that kind of fluctuation. So you don't have, so in plants, it rains sometimes and it doesn't rain sometimes, and that's a very large difference in water availability. In animals, there's no large difference from one time to another of availability of food. And so most dynamic energy budget modeling neglects that. Since you do have to do the differential equations numerically, there's no reason why you couldn't include that, and we eventually will. That actually probably makes a very large difference for the physiology. So one of the things that I've learned in the physiology model is that if there is no environmental stochasticity in relative humidity or rainfall, so if instead of it raining and then being dry and then raining again, you had controlled situation in which there was a steady drip of rain always at the same rate all the time, then the amount of water available to the plant doesn't seem to depend on the soil type. We got that result recently, and as I've looked at the model more and talked about it with people more, I believe that's really true for that case, but I think it's not so true for the case where rainfall is actually sporadic, because there the amount of different soil types to hold on to water while it's raining a lot makes a big difference. I'm not exactly sure how that works, but I think the clay soil is probably better for water availability to the plant than the sandy soil is.
Would that be due to hydrogen bonds with clay?
No, I think it could have a little bit to do with that, but I think it has more to do with the fact with pore sizes. Pore sizes in clay are really small, so once you get water into a pore, it takes quite a bit of water potential to pull the water out, whereas with sand, bigger pores, the water moves out more quickly. So when it rains hard, the clay fills up with water and it doesn't drain out very quickly, so it remains available to the plant longer. The sand just drains the water through. And so after it stopped raining, the sand goes back to where it was before it started raining faster than the clay does.